Principles of Inheritance and Variation, Session 20. In this module, we will learn how there is a parallel behavior of chromosomes and genes.
Also,
Chromosomes and
both
genes
chromosomes and genes segregate during the process of gamete formation, and only one of each pair is passed on to a gamete.
But
However,
they
they differ in the way they segregate.
at the time of meiosis.
In genes, independent pairs segregate independently of each other, while in chromosomes, one pair segregates independently of another pair.
Dear children, this figure will help us to explain the Mendelian principles of segregation of factors in terms of movement of chromosomes during the cell division called as meiosis. In meiosis, we know there are two stages, meiosis 1 and meiosis 2. In meiosis 1, there is segregation or separation of homologous chromosomes. And in meiosis 2, there is segregation of the chromatids taking place. This picture, if you can see, this cell bears a homologous chromosome. A homologous chromosome, one which is received from a father and one which is being received from the maternal parent or the mother. Each chromosome comprises of two chromatids, two arms called as chromatids. And these bears, each homologous chromosome or each chromosome bears an allele, capital R, and the other chromosome bears small r, capital R and small r. So here we can see in the first phase, that is metaphase 1, the homologous chromosomes are aligned along the metaphase plate. And these homologous chromosomes, which bears a pair of factors, and they separate in the anaphase 1. So this is the anaphase 1, in which the homologous chromosomes are getting separated. And in the next phase, we can see that is anaphase 2. In the anaphase 2, the chromatids get separated so that ultimately in the gametes, each gamete is receiving one chromosome along with one allele. Is it clear? So here we can understand the segregation of chromosomes with respect to segregation of gametes. Let us now study the behavior of chromosomes during cell division. In the anaphase stage of meiosis 1, a set of chromosomes align along the metaphase plate. Here we can see a long orange and a short green chromosome and a long yellow and short red chromosome aligned at the same pole. Now let us consider another set of chromosomes aligned along the metaphase plate. During the anaphase stage of meiosis 1, there is a long orange and short red chromosome and a long yellow and a short green chromosome aligned along the same pole. Now during the anaphase stage of meiosis 2, we can see the orange and green chromosome segregate together in the cell on the left, while the orange and green chromosome segregate together in the cell on the right. Sutton analyzed the segregation of chromosomes during meiosis and concluded that chromosomes have individuality and they occur in pairs, while members of each pair contributed by each parent. He also concluded that the parental chromosomes segregate from each other during meiosis, and the distribution of the parental and the maternal chromosome in each homologous pair is independent of the other pair. Both the scientists Sutton and Boveri were of the opinion that the pairing of and separation of a pair of chromosomes would result in the segregation of genes or factors carried by them, and their hypothesis on chromosomal behavior came to be known as the Boveri-Sutton chromosome theory. In fact, Sutton combined the hypothesis of chromosome segregation and Mendelian principles and called it the chromosome theory of inheritance. Thank you.